Hi, my name is Daniel Badie, and I'll be explaining to you how in 2004 these three scientists came together to win the Nobel Prize in Chemistry for their discovery,
Ubiquitin-Mediated Protein Degradation.
Let's dive deeper into the research. Israeli biologist Aaron Chekanover, Hungarian-born Israeli biochemist Avram Hershko, alongside American biologist Erwin Rose, all contributed to a series of groundbreaking experiments on ubiquitin-mediated protein degradation. In the 1980s, these three scientists began to work together in Philadelphia at the Fox Chase Cancer Center. This was where they began a series of experiments that uncovered the knowledge behind the ubiquitin system and how the cells regulate the presence of a specific protein by marking unwanted, denatured proteins with a tagging system using polypeptide ubiquitin. But first, let's talk about some proteins. Proteins work together as they're essential for the human body. There are thousands of proteins in each cell, all performing various tasks at the same time. A healthy protein can become damaged by mutations, denatured by a virus, or oxidized by oxygen, resulting in becoming a non-functional protein. Proteins can't survive forever when exposed to a warm environment, as some proteins are denatured within minutes. So the ubiquitin system is called to work every second as it identifies and replaces any damaged proteins. Without the ubiquitin system, we would resemble a rotten steak, as humans can't mitigate protein degeneration by staying in cooler environments. Damage to a healthy protein can be due to heat, radiation, or oxidizing by oxygen. The ubiquitin system plays a large role in our body as a healthy protein becomes damaged and becoming non-functional. This targeting of a denatured protein occurs via the E2 ubiquitin conjugating enzyme and the E3 ubiquitin ligase. Ubiquitin is involved in terminating mechanisms for many systems in our body. When phosphorylation occurs with serine, threonine, and tyrosine, ubiquitin is targeted by specific lysine residues. When a single protein is connected, it is monoubiquitinated. When multiple proteins are connected, it is polyubiquitinated and forms a polyubiquitin chain. How the building of the polyubiquitin chain occurs is by the E2 and E3 complexes binding to the denatured protein. E1, the ubiquitin activating enzyme, binds to lingering ubiquitin floating around and then transfers it to the E2 enzyme that conjugates ubiquitin covalently to the damaged protein. This process of E1 finding ubiquitin and binding to the E2 enzyme is done multiple times as the ubiquitin subunits are bound to ubiquitin substrates. E1 continues to collect ubiquitin until it is done binding the necessary polyubiquitin chain. Proteins in the body are constantly being damaged and recycled. The ubiquitin system consists of 2,000 special protein molecules that identify degraded, damaged, or unwanted proteins that could lead to the development of diseases and disorders. This tagging attracts proteasomes that recycle these proteins. The recycling of unwanted proteins occur at all times in the body, regenerating 5% of your body's cells daily. The same 20 amino acids constitute all proteins, but are arranged differently to express special functions. So the proteasome will target the ubiquitin-targeted proteins for their recomposition to functional proteins, as the proteasome essentially cuts the protein apart to isolate amino acids. This polyubiquitin chain binds to the 19S complex of the 26S proteasome, making the substrate misfolded and degraded, while ubiquitin is recycled into the environment. Ubiquitin-labeled proteins are deconstructed into amino acids. It's like having individual letters and making sentences with them, or having a pit crew that repairs your car. But what happens when we get older and these systems stop working as well? This can be seen in the accumulation of tau proteins in the brain for Alzheimer's patients, as well as patients with cystic fibrosis, and when cancer cells begin to reproduce. For my biotech company, I would focus on the progression of cancer treatment research when targeting the ubiquitin pathway. Advancements in the technology could be done by re-engineering the different ubiquitin complexes. Each of the E1, E2, and E3 subunits could be genetically modified to exhibit an increased level of binding for drug development. Or instead of repurposing amino acids, you could engineer it into a modified protein. There has been an extraordinary amount of research that show radiation treatments aid in the shunting of unwanted cancer cell development. With surface-guided radiotherapy, you could activate ubiquitin for tumor treatment to stimulate protein pathways in specific areas that will inhibit tumor growth. Lastly, I would re-engineer the ubiquitin system to particularly target proteins that promote unregulated cell division. By degrading these proteins, it would prevent cancer cells to reproduce, and it would remove any damaged proteins.